Okay, so another set of important compounds in our body are going to be fats or lipids. And so lipids in our body is going to have three main roles. One is what we always think about, of course, which is storage of energy in the form of fat. And another is our cell membrane components. And then the third is they're going to be important in cell signaling. Okay, so we have our fats, our stored lipids, we have our cell membrane lipids and form a lipid bilayer. Right, so outside the cell and inside the cytoplasm is a lot of water. So you have your hydrophilic regions exposed to water, and then inside the membrane, you have your hydrophobic regions or your fats inside the membrane. And of course, in signaling, we have a lot of hormones that are made up of components of fat. All right, so lipids are going to be important in humans. And the major lipids we have are called fatty acids. And these are going to be esterified or linked to a glycerol backbone. And these are going to form our triacylglycerols or our tags, which are also called triglycerides as another common name. Or it can become a phosphoacylglycerols. And so again, we have our glycerol, which is a 3-carbon compound. So this is glycerol. And it forms sort of the backbone of our fats, especially our stored fats. And so on each of these oxygens associated with the carbon backbone of glycerol, you're going to get a fat. And so this whole molecule here is a tag. All right, so sphingolipids, we'll talk about in just a moment, are basically a fatty acid attached to sphingosine. Sphingosine is an 18-carbon amino alcohol compound with unsaturated hydrogen carbon chains, but we'll talk about that in just a moment. We have our glycolipids. Glycolipids are sugars attached to the lipid hydroxyl groups. So remember, I talked about glycosylation. It's going to be important proteins, but also lipids sometimes have sugars attached to them. We do have polyunsaturated fatty acids that are going to be important for the immune system, and they're called eicosanoids. And these come from arachidonic acid. Which comes from our essential fatty acids. And of course, we have cholesterol. Cholesterol is going to be important for many things, including your membranes. You do have cholesterol embedded in your membranes. It's very important to the structure of the lipid bilayer. You also make bile salts, which are very important for the digestion of fats, and steroid hormones. So the cholesterol forms the nucleus or the backbone for a lot of the hormones that we make. Okay, so let's just talk about fatty acids. Fatty acids are the simplest lipids. And they're going to have a long hydrocarbon chain. So here's our long carbon hydrogen chain. And in humans, in our bodies, the most common are going to be 16 because we make palmitate. You're going to find out when we talk about the liver is going to make palmitate. But stearate, carbon 18, is also very common. You can have even carbon 16, 18, 20, and those are going to have a carboxyl, the COO minus at the end, and a methyl group at the other end. When you hear about omega fatty acids, this is your omega carbon down at this end. So here's omega. And then when you talk about the conventional way of numbering your fats, your carbon 1 is up here at the other end of the omega. Saturated means no double bonds. So saturated with hydrogen. Unsaturated means you have one or more double bonds. And these can form kinks, especially in your membrane, your lipid membrane. So here is just a series of fatty acids. We'll walk through these a little bit. We have our 16 palmitic, 16 carbon acid is going to be formed by our liver. And then we also have stearic acid, which is going to be 18 carbons, which is also common. Cis is natural. Trans is artificial. And this is with respect to bonds, double bonds. So trans is going to be formed in the actual process of generating some food product, and you get hydrogenation of a polyunsaturated fatty acid, and it causes a trans bond. Another thing to keep in mind is as the length increases, the melting point increases. So the longer they are, the more hydrocarbons they have, the more it takes to melt them. Now just a little bit about conventions. So again, we have our first carbon over here, carbon number 1. We have our omega carbon starting over here. The second carbon is called the alpha carbon, okay? The beta carbon is the third carbon, so keep that in mind. When you have a double bond, you're going to see this 1 delta and some number passing over that. So this means you have an 18 carbon lipid with 1 double bond starting at position 9. So the double bond is between 9 and 10. So you see here we have 9, and here would be 10. And so this is an 18 carbon with 1 double bond between position 9 and 10. Here we have an 18 carbon with 3 double bonds, and they start at positions 9, 12, and 15. Therefore, the double bonds are between 9 and 10, 12 and 13, 15 and 16. And you see that they're all in the same length, so they're cis. The trans double bond would, of course, be going in this direction. This is a trans. Ooh, sorry. Here's your trans. Goes in this direction, and this guy's your cis. Arachidonic acid, which again comes from our essential fatty acids, has a double bond, has 4, and these are the positions. It's also called an omega-6. So you see this omega by each of these. I'll change the color of those. So our omegas, omega-6, omega-3, and omega-9. What that's referring to is you start on the omega side. This is omega-1. Here's omega-3. Here's omega-5. Here's omega-7. And here's omega-9. And on this one, you have omega-1, omega-3. Here you have omega-1, omega-3, omega-5. This one would be omega-6. And so basically, when you count omegas, you count from the opposite end of your COO group. That's your 1. And then you go to your double bond, and whatever omega that is, that's what you call it. So an omega-6 has a double bond start from the omega end starting at omega carbon 6. All right, so fats are acyl glycerols, which is basically going to be our glycerol backbone with our fatty acids. And so triacyl is going to have glycerol and 3 fatty acids. And this is stored fat. Now, from this basic structure, you can do many things with this. So here again, we have our backbone. This is our glycerol backbone. And then we're going to have our 3 different fats. So here's our fat 1, here's our fat 2, and here's our fat 3. And they're all bound to our backbone. So if you have a phosphate group attached to that, that would be a phosphorylacylglycerol. And the phosphorylacylglycerol would have, again, oops, sorry, wrong one. We have our glycerol. So here's glycerol. You still have your fat. So here you have your fat. And now, instead of a 3rd fat, you have this phosphate added. And in this case, phosphate is linked to choline, so it's called phosphatidylcholine. But it's still the same glycerol backbone. You just swapped out one of those fats for a phosphate group, and in this case, attached to a choline. Phosphatidic acid is shown here, in which you have just a phosphate group with nothing else attached. So what that means, phosphatidic acid is going to be a building block for many different things to come in and attach to that oxygen. And shown here is choline attaching to that oxygen. Now, sphingolipids are going to be derivatives of a compound called ceramide. All right, so here we go. We have ceramide. And ceramide is basically going to be sphingosine and a fatty acid. Now, what's a sphingosine? A sphingosine is going to be serine and palmitate. Remember, palmitate is that 16 carbon. Okay, so here we have our serine now. So this is not our tag. This is not glycerol as a backbone. This is now serine being that backbone. And then serine is going to be attached to our fatty acid palmitate, and that equals a sphingosine. And then you're going to have our sphingolipids here. And a sphingolipid is basically serine plus 2 fatty acids. So sphingosine is going to be serine plus palmitate, which is basically 1 fatty acid. A sphingolipid is serine plus 2 fatty acids. And a ceramide is serine plus palmitate plus another fatty acid. So that's why they say a sphingolipid is a derivative of ceramide. Now, what's important is you're not really saying what this fatty acid is necessarily. So it could be an 18 carbon, 20 carbon, something longer. But they have serine as a backbone instead of glycerol. And so you have sphingomyelin, which is going to be important in cell signaling. You have other famous ones are our galactocerebrosides, which are skin lipids. We have our oligosaccharides, gangliosides, which an example is sialic acid. And sialic acid is a receptor for influenza, for example, and it's also going to help your cells adhere to one another. It's a cell adhesion function. And then finally, let's just talk a little bit about steroids. Steroids have 4 rings, right? So you have your A, B, C, D. So that's considered your steroid nucleus, those rings. And they're not very soluble. They are going to be, of course, a precursor for our hormones and our biosalts. So cholic acid is an example of a biosalt. And you'll find this on the surface of my cells. Or micelles are those lipid particles that you make in your lumen of your gut when you're eating. So the lipids emulsify into these particles called micelles. And cholic acid is the biosalt that helps emulsify them and make them more soluble as they move through the lumen of the gut. And so this is an important function of cholic acid, which is an actual cholesterol molecule. And then, of course, we have our testosterone, which is a hormone, and estradiol, estrogen, another hormone, and those all have steroid nuclei.